So hi everybody, and thank you for your attention. While I'm going to discuss how population genetics is really important to consider in designing solar facilities, especially when you want to have solar facilities that have the least amount of environmental impact. So to start off with, I just want to take a moment to recognize that Michigan State occupies the ancestral, traditional, and contemporary lands of the Anishinaabe, which is the Three Fires Confederacy of Ojibwe, Ottawa, and Potawatomi peoples. In particular, the university resides on land that was actually ceded in the 1819 Treaty of Saginaw. So to start us off, as we've seen the demand for renewable energy growing and growing, solar energy is one of the more appealing forms to developers simply because it's really, really clean compared to other forms of alternative energy such as like wind farms. And due to that, we can kind of see that there's been an exponential increase in solar development. This involves like all three forms of solar development, which is like residential, non-residential, and utility scale. But this is just talking about the photovoltaic cells, which is kind of like what people think of when they think of solar energy. So those cells that just kind of sit facing the sun and convert the solar rays into energy. The only thing about utility scale solar energy in particular is that these are really, really big facilities, and they tend to have pretty large and what are often unexpected ecological impacts. And of course, as these things tend to go, there is one type of ecosystem that has the highest solar potential and also happens to be quite sensitive. And those would be our deserts. So most people don't really think twice about deserts, especially since a lot of us are kind of living in the Midwest, and deserts are not something we see every day. But deserts are actually really, really important to many, many people worldwide. And actually, roughly about 35% of all terrestrial area is classified as arid. And within that, 10% of that is just in North America alone. So a lot of people who are not really familiar with deserts tend to think of them as like barren wastelands, kind of like what you see here in this lower quadrant, which is actually the Bonneville Salt Flats, which are outside of Salt Lake City. However, a lot of times deserts actually serve as really crucial havens for both native wildlife and really cool things such as migratory birds. And we see an example of that here, which this is the Stillwater Wildlife Refuge in the Great Basin Desert. And this is like really, really crucial habitat for this region. On top of that, these really fragile ecosystems actually tend to contain really incredible biodiversity, where you might see up to like 70 species per hectare in some areas of the Eastern Mojave. This kind of creates this intersection of arid lands and solar energy. Specifically, the arid lands of the desert Southwest in the US have some of the highest solar energy potential in the entire world. Some other things that make this really appealing to developers is the fact that most of this land is publicly owned, aka owned by the government. And where there are people living out in the desert, they tend to be in clustered, really highly populated cities with lots of open space. Separating them, you can think of places such as like Las Vegas, Phoenix, and kind of like LA. So this creates this really interesting catch-22 between developing solar energy being great for providing alternative sources of energy to get us away from petroleum use or coal use, and preserving the desert ecosystem. Previous studies have already shown that these large facilities tend to alter wind patterns, rain. We've observed these, but we don't really know much about kind of the short and long-term impacts of these changes. And this is really important to think of specifically because solar facilities are all built to be taken down. So a lot of them have decommission dates that fall within 20 to 25 years after they are opened. It's really, really important to think about how these solar facilities are impacting the desert communities, especially because desert communities are like notorious to recover from any type of disturbance. A lot of times it can take up to 76 years for a region to fully recover once there's been some sort of disturbance. And this can even be something just like a road being built. So really, really long timescales to recovery. So kind of narrowing down further, I specifically am interested in the Mojave Desert and this region of the US, which is down kind of spanning southern Nevada, California, Arizona. is actually considered a biodiversity hotspot. So it's thought that not all species, and of those, especially plants, have been characterized in the Mojave Desert, with many of these plants actually only being rediscovered by botanists in the past decade or so. Over 50% of the Mojave is actually contained within California, which is nice in one aspect because California tends to be more interested in things like renewable energy and species conservation. And according to the California Native Plant Society, 25% of the region's species are actually endemic to the state. So some examples of those would be this showy Clark Mountain green gentian, which is actually only found in one spot in California on the limestone substrates of Clark Mountain. We also have something like Thorn's buckwheat and the red rock tar plant. So conserving these rare plants poses a unique challenge because most of the basic biology of many of these species, such as distribution and life history information, is completely unknown or extremely poorly documented. So to narrow down a little bit further, the region that I feature in my study is outlined in this circle here. So it's the Ivanpah Valley. But before going any further, I would like to recognize that the Ivanpah Valley is actually within the ancestral lands of the Nuuvi as well as the Niue people. And I personally would like to offer gratitude for the land itself and also for those who have stewarded it for generations. And for this opportunity, I had to study, learn, work, and just kind of be in community with this land. So to further introduce this area, since I'm talking about solar facilities, the facility that I get to study is the Ivanpah Solar Electricity Generating Station. So this facility began construction in 2010 and officially opened in 2014. At the time of its completion, it was actually the largest solar facility of its kind, which this is specifically a concentrating solar power facility, which is designed with a bunch of mirrors that all kind of point the solar rays up to these crazy collection towers that superheat a liquid. Older facilities such as this one contain water. Newer facilities actually contain molten salts, and that superheated liquid generates steam that turns turbines. When it was completed, it was the biggest in the world, and even to today, it's actually the highest producing facility, which it creates enough power to power over 140,000 homes. So another cool thing about this facility is that when the developers actually started building this facility, they elected to not grade the ground underneath most of the mirrors to try and save the plant life. So you can kind of see that a little bit here in this picture, where there's mirrors here on the edge, and you can see that there's still a whole bunch of, bushes and things like that within the facility. So another thing about this facility is they kind of tried to make allocations for rare plants that they knew were in the area. But it is important to note that when they actually went out and sited this area before constructing the facility, it was a dry year. So of course, what ended up happening is after construction began, there was a wet year, and a lot of rare plants in the facility that had been dormant when they were siting it popped up and essentially ruined the developer's day. So one of these plants, which is the plant of my study, is the Mojave milkweed. So when this plant appeared, some efforts were made to relocate it, but that ultimately failed. So what ISIGs actually ended up doing is they altered the design of the facility itself by setting some of the mirrors back into what turned out to be these kind of like halo-like structures when you view it from above. So there's just little pockets of space within the facility, and I mean little, kind of just sprinkled throughout the facility to kind of try and limit the impact on these milkweed. So what that did is it left the milkweed in the area directly undisturbed, but now they're actually also growing in kind of a bubble within the sea of mirrors. So it's kind of unclear how this is going to affect the milkweed, especially because it tends to be localized to drainages and washes, and there are generally changes in the water patterns within facilities. And we've seen cases where a wash has been completely wiped out and plants that we knew were in that region are no longer growing. So another cool thing about this plant is it's actually an important food source for some of our pretty charismatic insects, such as monarch butterflies, and they mimic the queen butterfly. But this also has been observed to be eaten by jackrabbits. And there's also like sphinx moths and lots of cool like milkweed beetles that rely on this plant. And it's also currently classified as common but declining because these plants featured in my study are actually the only ones that we've categorized as growing in California. So this kind of sets us up to kind of approach some really interesting questions. So first of all, since next to nothing is known about the basic biology, even less is known about the population genetics of these desert plants. So we kind of need to determine what the current population structure is of the milkweed before we can even conserve it. These are questions such as, are there distinct populations? Is this facility, which is a 3,500 acre facility, large enough to overlay an entire population? And our second question is, so is there unique diversity actually within the facility that we should take into consideration? So this kind of information would be really valuable to solar facility designers because the kind of unique features of ISIGs, especially the fact they didn't grade the ground, kind of comes with a financial burden. It's a little bit more expensive to maintain these facilities. But if we can actually show that these techniques were sufficient in, preserving what might be a really, really important group of these plants, this might help sway designers of future facilities to not grade the ground beneath their mirrors. For our field sampling methods, we are actually went out to the field twice. Collection occurred in spring 2015 and again in fall 2018. And we took samples from as many plants that were large enough to sample. Some of them were just seedlings when we were out there, so we did not collect those because that would have killed the seedling. So that was about 100 samples in 2015 and 137 samples in 2018. And for all plants that we saw, we recorded their GPS coordinates. So this includes everything that we were able to sample as well as those that were there, but we were unable to sample. And to get an idea of kind of what where the populations are in space, here are our sampling locations. We have kind of three natural areas, and the one area within ISIGs, which is all of these kind of like black smeared dots. The entire area is kind of outlined in this oval. So you can really see the difference in the spread of those plants versus the other areas. So it is also important to note that two of these areas, which is Bobcat, this one way down south, and Excelsior, which is this green group, were only sampled in 2015, but we were able to get a whole bunch more samples from Umberchi, which is like directly north of ISIGs, and within ISIGs itself when we resampled in 2018. Okay, so once we got our samples, we dried them out, and then I extracted DNA from them. And then we prepared them using the best rad technique, which essentially uses a restriction enzyme. In this case, it was PSTI for any of you that are interested. And it just like randomly chops up the genome and allows us to subsample in smaller fragments. And then we sent it off to be sequenced. And once we got that data back from the sequencer, I aligned the reads to the Asclepias syriaca genome, which is common milkweed, and then did SNP discovery in the program stacks. And that gave us a little bit under 11,000 loci across all of our individuals. And then ultimately, after a bunch of quality control, I ran several analyses to determine what the population structure looks like. So answering that first question, and then also to kind of get an idea of what the diversity of that region is. Okay, so our results for population structure is that we do get 4 distinct clusters. So those 4 sites that we have do end up coming out as 4 distinct populations. So we performed both a structure analysis as well as this is a DAPC clustering analysis, and it gave us the 4 clusters. And what's interesting is while 4 clusters was best supported under the 3 cluster model, what would end up happening is Umberchi, the blue group, always came out first, but sometimes under K equals 3, Excelsior and ISIGs would be combined, and sometimes ISIGs and Bobcat would be combined. And this is also, these kind of like groupings of sites into populations is supported by our FST values. So these populations are pretty well differentiated. So to kind of further tease out these genetic relationships among individuals and populations, I did what's called a minimum spanning network analysis. And essentially what that does is it compares like pairwise genetic differences between individuals and then links them together based on these differences. So the individuals that are farthest apart, such as these blue individuals down here and these orange ones up here, have a pretty great genetic distance between them. So they're, from a genome standpoint, the most dissimilar. So now that I've oriented you a little bit, I would like to point out what is perhaps the most interesting thing about this figure. So our I6 population, again, the black samples, is actually central to the other populations, which indicates that it served as an ancestral source of plants for the other populations. So this is actually supported by some of these other individuals from other populations clustering kind of here within I6. Now, this might be a little confusing because this looks a little bit different than what we see in our DAPC analysis. But what I'd like to point out is that DAPC analyses, so these allow for kind of like multiple dimensions to be considered, such as certain alleles might contribute much more to the population structure than others. So it's much more kind of fine scale. And this minimum spanning network analysis is much coarser in that it's only pairwise distances. We see kind of like the history of like these individuals sharing a lot of their genomes with ISIGs, but they still are reasonably clustered within what we see in our DAPC. Okay, so that answers our first question. So to our second question, we pulled out private alleles. And what we saw in private alleles by population in 2015, which is when we had sampling from every single site, we had 2,500 private alleles, and many of them were actually found in a single individual. And it's important to note that milkweed can be clonal. So we actually had clones removed from these data sets when we did our private allele analysis. So we did this again when we combined both sampling years. So that would be when we had a whole bunch more samples from Umberti and ISIGs. And kind of the expectation for that would be that there would be an overall decrease in private alleles simply because the two populations that we sampled again were geographically closer to one another than anything else. So we figured that they would actually share many alleles. And we actually kind of see the opposite trend here. So ISIGs really has the largest proportion of private alleles when we combine both sampling years. So what this kind of implies is that when we went back and resampled, we sampled a whole bunch of plants that we didn't initially know were there. So we got a better picture of the fine scale genetics in that area. And this showed us that there's actually a lot of cryptic diversity kind of nestled in ISIGs. Now that I've given you all that information, let's again revisit the questions. We did see that the milkweed and the Ivanpah Valley all cluster out into individual populations. Again, the question of, is there unique diversity within ISIGs? Again, yes, there are 4 distinct populations. And also we see that ISIGs does contain quite a bit of unique diversity. And the facility, since it does in fact overlay an entire population, it is important to note that the halos plus the fact that they didn't grade beneath the mirrors proved to be really important modifications to maintain the plants, because otherwise if they had graded everything, we would have lost an entire population and all of that diversity in the region. So some future directions that I am thinking of going in is maybe coupling this genomic information that we have with some ecological data such as rainfall, herbivory metrics, and temperature for better biological context for this region. And then it might be interesting to model some of the potential altered temperature and water regimes to kind of maybe predict what the ultimate ISIG's influence is going to be on the milkweed in the area. So to wrap everything up, the milkweed within ISIGs is a distinct population and has quite a bit of unique diversity. This kind of just adds more fuel to the idea that deserts are super diverse and they're really, really interesting and important, and we need to put resources into learning more about them and preserving them. And since solar energy is continuing to grow, it's going to be important to have these considerations in designing facilities that both mitigate the effects of development and help deserts recover more successfully after they're decommissioned. And with that, I would like to acknowledge my lab members and some of my collaborators, including Steve Grodsky and Kara Moore O'Leary, as well as the MSU EAB program that provided some funding for all of this. And with that, I will take any questions that you all may have.